So what we are talking? We were talking that. Okay. So we are not only this thing going through with theoretical things, we are also doing hands-on research, isn't it? So we will do a little recapitulation that what exactly we are trying to do. Okay, then we will go to this. Okay, so this is a database which will give you a lot of info on the beginning of biomedical research. So we were discussing, just hold on. We were discussing this. We were discussing what? We were discussing molecular basis of vitamin D and linked endocrine regulations. Okay, so let us consider that we are discussing this particular topic. So how we will start our journey? So we can start it like this. So this is our topic of interest. So one by one we will be doing things. Okay, first of all we will learn from theory of vitamin D3 and parathormone. Okay, we will learn a little theory on it, because what happens, as we know that absorption of vitamin D3 is linked to parathyroid hormone. Okay, so in order to indicate that, we will say okay, we will come back to this thing later. So here what we will do is we'll just do a little theoretical study that what exactly are there already. So we are basically interested in the endocrine regulations of vitamin D3. Okay, so this is our theory. This is one theory. Some of you are already aware of it, that the idea is to identify vitamin D3 deficiency and elevation of parathyroid hormone, how these two molecular events are connected in a genetic disease that is called thalassemia minor. So this is a case study that was been published long back in 2012. So in this study, what we figured out that beta thalassemia minor, which we already discussed, that which is a what? Which is an yeah, just hold up. Yeah, beta thalassemia minor is a genetic disease which can have two different what you call aspects. One is beta thalassemia minor and beta thalassemia major. In major cases, which is basically a recessive homologous mutation, which leads to complete this thing hemoglobin deficiency. On the contrary, if it is a heterologous mutation, it will lead to low hemoglobin concentration but increase in the number of RBC. Now increase in number of RBC in contrast is connected to low total cholesterol in the blood. Okay, we discussed these things. So low total cholesterol means what? Low total cholesterol in turn is related to low vitamin D deficiency. Okay, low vitamin D concentration. Why? Because cholesterol is the what you call raw material for the formation of vitamin D3. Okay, these are multiple different things that is going on. Okay, so here also you can see that cholesterol is also present where? In the plasma membrane of RBCs. Okay, so since RBC numbers are increased in thalassemia minor, remember RBC number is increasing, at the same time total hemoglobin is also decreasing. So anemia is there, but total number of RBCs are increased. Okay, So in this case what happens, the cholesterol concentration in the blood decreases, which also leads to or may lead to deficiency in vitamin D3, because vitamin D3 is nothing but a derivative of cholesterol. Now how vitamin D3 acts? Vitamin D3 receptors are present on the this is vitamin D3 receptor, which is present on the surface of all kinds of cells. Okay, the receptor is having basically two domain. One is extra cytoplasmic domain and another is cytoplasmic domain. Okay, there is also a transmembrane domain. This is the transmembrane domain. So vitamin D comes and binds in this, and eventually a G protein coupled reaction starts, which helps the cells to accumulate calcium by opening the calcium channels. Okay, so tell me if anybody is aware of the calcium channels. Who can tell me if you have some idea of calcium channels? Huh? Anybody have some idea on calcium channels? What are calcium channels? Where you can find it? So calcium channels are basically present on all cells, more precisely on cardiac muscle. Okay, so what exactly they do is, consider that this is your calcium channel. So what they do is Ca+2 channel. So what they do is they enhance the influx or outflux of calcium to and from what we call cells. Okay, now calcium, who can tell me what is the cellular function of calcium? That means inside the body, what are the major functions of calcium? Can anybody tell me? So I'll give you some example that why calcium is needed. Calcium is needed because it is the factor 4 of blood coagulation pathway. Okay, this is one thing, blood coagulation pathway. Okay, and calcium is also needed because it helps in muscle contraction. Ca+2 helps in muscle contraction via actin myosin filaments. Through the sarcoplasmic reticulum. Okay. Okay, understood. So these are the major functions of what we call calcium. So these are not only this, there are multiple different as well. Some calcium ion is also needed for proper folding of proteins. Some proteins are there which are calcium binding proteins. For example, okay, the third point we can write. Third point is regulation of calmodulin. Okay, regulations of calmodulin. These are a group of proteins which are responsive to calcium concentration, and they perform multiple different activities. Okay, so these are the different aspects that why we should learn about our topic, molecular basis of vitamin D linked endocrine regulations. Okay, so we can change the topic later on. So we have done some hands-on as well. So today we will do a different hands-on. For that we will share with you some tools. Okay, so one by one I will be sharing the tools. First and foremost, I will share one tool which is prepared by a university which is called, the name of the university is Harvard University. Okay, so we will use a tool today which is developed by Harvard University genetics department. Okay, and we will have a background check as well, that why this particular tool has been generated in the first place. Okay, so we will be using two tools. We will be using to analyze impact of mutation on vitamin D3 receptor. Okay, so can you remember what is the vitamin D3 receptor? So this is our agenda, so we'll write it down here. So today we'll learn what today we'll learn what the tool to analyze the impact of mutation on a vitamin D3 receptor. This is the task of today. So we'll give you a recapitulation that what exactly happens. So if the structure of the VDR receptor is altered, so what will be the effect that we shall start to find out. So can anybody tell me what will be the effect of the mutation in the VDR or vitamin D3 receptor? What it will lead to? What will happen?
So it can lead to improper binding of vitamin D3.
Okay. So when you say generic terms like improper binding and so you exactly cannot say that whether it is enhancing the binding or reducing. So what exactly happens is any kind of mutation. Let us discuss mutation first. So what happens? We already have discussed that there are multiple types of mutations. We will consider these today. Not all, at least we will consider these 3 mutations today. Okay. So one is called missense mutation. Missense mutation. Okay. Then there is called nonsense mutation. Okay. Then there is again frame shift. Sorry for this clerical mistakes. Frame shift mutation. So all these mutations we will see today. So one by one, a little bit recapitulation. You might have already learned these things. So missense mutations is nothing but a mutation where where a single nucleotide polymorphism takes place. Okay. So this is nothing but an SNP, or we shall also call it single. How many of you have heard this term? Single nucleotide polymorphism. So this is one thing we will discuss today. So it exactly what? It is nothing but a single nucleotide will be changed and it will eventually lead to some kind of impact. So in nonsense mutation what happens? Formation of a stop codon takes place. Formation of a stop codon. Who can tell me what is stop codon in a protein? Okay, you take all these notes, take screenshots. It will help you for your GATE, NET or all kinds of biomedical research based or biomedical based entrances. It could be DBT entrance, it could be CSIR NET, it could be GATE also. So for all kinds of students these informations are needed. So what it will do is nonsense mutation, it will lead to formation of a stop codon. So there are 3 stop codons. So if we start discussing, we will discuss many things. Who can tell me what are the stop codons?
So stop codons are those codons like UA, UAG or and UGA, where the amino acid protein synthesis stops.
Yes. Yeah. These are the 3 stop codons. They will appear due to the mutation. So what would happen? Consider this. So what is the impact that you understand first here? So we are discussing this. Okay. So consider that this is your protein. Okay. Native protein. Okay. This is a native protein. Then a nonsense mutation takes place. A nonsense mutation takes place. Okay, this nonsense mutation, know what it will do, you know. Okay, so this nonsense mutation takes place, and what it will lead to? It will lead to formation of a protein, the same protein, but it will get terminated. Okay, so can you see a smaller protein? So premature termination of protein will take place. Premature termination of protein. Okay, a smaller protein will be formed. Okay, this is the impact of nonsense mutation. So let us see what is the role of missense mutation. in missense mutation, again we will take one more protein structure. Let us say this is the structure of a protein, and what is the impact of nonsense mutation we will see. Sorry, missense mutation. Okay, so say native protein, okay, so native protein, and this is missense or frame shift, both frame shift mutation. Okay, so see the impact of these, just hold on. So this is the impact, and it forms what? It forms a completely distorted protein. Okay, the protein will be formed, but it will be completely distorted. Distorted means there will be no structural symmetry. Okay, so these are the impact of these what we call as mutations. Okay, so frame shift alter, it will lead to this. Okay, so now we'll go to our this thing tool. Okay, so in order to identify the impact of these three kinds of mutations, missense mutations, nonsense mutation, and frame shift mutation, we'll use a tool. The tool is produced by, or tool is developed by Harvard University Genetics Department. Okay, Harvard University Genetics Department. Simulation toolbox. Okay, so we'll study this today. Okay, so this will study to analyze the impact of mutation on vitamin D3 receptor. Okay, now I want to discuss a little bit more on this tool. So interesting feature of this tool is that, it is based on an algorithm called, it's a machine learning algorithm. The tool is based on a machine learning concept which is called naive Bayes algorithm. Okay, so we'll study this today. Okay, so the tool is based on a particular kind of algo which is known as naive Bayes algorithm. Audible? No? Audible, visible, everything? Say yes. Okay. Okay. So we'll go to the toolbox now.